Hello students, I am Dr. Neeraj Gupta and is working as Assistant Professor in the Department of Biochemistry of Himalayan Institute of Medical Sciences. Today I will talk about sunlight and vitamin D, focusing on vitamin D production and its various photoproducts with their biological actions. During exposure to sunlight, the wavelengths of 290 to 350 nanometer, which comes under UVB radiation, penetrate into the skin layer and activates the double bond of the D ring of 7-dehydrocholesterol and open it to form pre-vitamin D3. The pre-vitamin D3 undergoes rearrangement of double bonds in its structure to form vitamin D3, which is more thermodynamically stable and flexible. The vitamin D3 gets ejected out of the plasma membrane into the extracellular space and diffuses into the capillary bed in the dermis, where it gets bound to vitamin D binding protein. It will now be transported to liver for its first hydroxylation. In liver, it forms 25-hydroxy vitamin D3, that is calcidiol, and finally in kidney, it forms 125-dihydroxy vitamin D3, calcitriol, which is the active form. Now, a very interesting fact I would like to share is that during sunlight exposure, only 15% of 7-dehydrocholesterol is converted to pre-vitamin D3. The rest of it is converted to other photoproducts like luminestrol and tachysterol, which have no effect on calcium metabolism. Now, any further exposure to sunlight will result in photoequilibrium and vitamin D3 is converted back to luminestrol or tachysterol or is reverted back to 7-dehydrocholesterol. In addition, pre-vitamin D3 can also be converted to various suprasterols and also to 5,6-trans vitamin D3. And pre-vitamin D3 can be converted to toxysterols. These myriad of photoproducts produced during sunlight exposure have other biological effects such as regulating epidermal cell growth and reducing risk of skin cancer. Now, in one of the experimental study, the human keratinocytes were treated with different types of suprasterols, where suprasterol 5 and 6 demonstrated anti-proliferative property. Now, this slide shows percentage of cell growth as compared to controls. Now, here it can be seen that suprasterol 5 and 6 have significantly affected the growth of keratinocytes. The vitamin D shown here is taken as positive control. Now, what are negative controls? So, negative controls are particular samples included in the experiment that are treated the same as all other samples but are not expected to change due to any variable in the experiment. The positive control samples will show an expected result, helping the scientists to understand that the experiment was performed properly. Luminestrol is converted to 125-dihydroxy luminestrol and it has been shown to have some anti-tumor effects in skin. So no matter how much sun a human is exposed, there will be no intoxication of vitamin D. Besides, the phytoproducts generated may have additional benefits above and beyond than simply taking a vitamin D supplement or from dietary sources. I hope you have learned something new today and believe this information will arouse your interest in the health benefits of sunlight exposure. Thank you and keep learning.